Good afternoon. We are starting the first seminar, CIEN seminar. If you might remember, we ended the year talking about molecular mechanism of resilience, and we start the year with the same, exactly the same topics, a topic that we love. And we have today Ibai Diez. He's Ramon y Cajal from
Biobizkaia.
Biobizkaia and Mass General Hospital. We knew about him because he was working in the lab of Jorge Sepulcre, a friend of us, and he's going to talk today about well, the interface between imaging and genetics. So thank you very much.
Thank you very much for the presentation. And thank you very much for inviting me to give this talk. I am going to be talking a little bit about different neuroprotective mechanisms that we have to preserve the cognitive decline in Alzheimer's disease. No? I came a couple of months ago back to Spain with Ramon y Cajal. I am trying to start a lab in Biobizkaia, a research line, focus on trying to understand these protective mechanisms. No? So we are using a lot of in vivo analysis with neuroimaging and also both MRI and PET, and also we are using a lot of molecular imaging to trying to do disease models and try to understand what is happening in these resilient individuals. Also with plasma analysis, but lately we have been applying a lot postmortem analysis with omics to try to understand what are the molecular mechanisms of the things that we are seeing. And with all of this, I am a computational guy, so what we try to do is to integrate all these data modalities and try to estimate different potential targets that can mimic this resilience in the brain. So finally, we have some targets that we want to experimentally validate with cell cultures. So this is a little bit the idea of what I am trying to establish as a research line in Biobizkaia. And it's a little bit what I am going to present today and the things that we are going to do. So first, I will start with an introduction of the different neuroprotective mechanisms, and then I will focus on the research that we have been doing on resilience and resistance mechanisms. So not everyone at risk of Alzheimer's disease will develop cognitive decline. There is 1/3 of the people that go to our brains in autopsy that they have amyloid and tau levels, the hallmarks of Alzheimer's disease, equivalent to an Alzheimer's diagnosis, but they don't have cognitive decline. So trying to understand what are the molecular mechanisms of these people could help us develop maybe new and more effective treatments for these individuals. No? And when we speak about neuroprotective mechanisms, we know that the biggest correlate of cognition is synaptic density and neural density. So if we speak about neuroprotection, we have to speak about protecting these neurons. So we have to take into account also that in the brain we have other type of cells like glia, and that they play an important role in protecting the neurons. So with it, we will start to accumulate amyloid plaques. And for example, the microglia is doing is surveillance the brain. And when it finds some amyloid plaques, it will migrate to the amyloid plaques. And what it has the microglia is it can change the morphology and what they do. No? So it will change the morphology to an activated microglia, and it will start eating the amyloid plaques. What it happens, here we have the first neuroprotective mechanism. Some individuals, they have the ability to remove these amyloid plaques really quickly, maybe because they have a very efficient immune system or something like that. And then what we also have is intracellularly inside of the neurons, we start to accumulate tau tangles. And usually when you start to accumulate tau tangles, the microglia will change again to a phagocytic microglia, and this microglia will start eating the neurons. And here is where we find the second neuroprotective mechanisms, that it will be resilience. We have individuals that they have amyloid and tau, but somehow they don't decline. Maybe they can protect the neurons in some other ways or so on. So these are the two main mechanisms that I will be speaking about, resistant that will be avoiding the accumulation of amyloid and tau, and then resilience that is more how to cope with amyloid and tau to preserve the cognition. And then if the disease continues to advance, usually if we have a lot of neural death, this region of the brain is not going to work properly. So the last mechanism will be a cognitive reserve for neuroplasticity. There are some, the brain will create alternative connections to try to compensate for this damage. So why it's important to differentiate these neuroprotective mechanisms? Because depending on the stage of the disease you are, it might be more interesting to apply one mechanism or the other. For example, now we have these new drugs for Alzheimer's disease like lecanemab, donanemab, and so on, that they are very interesting as a resistance mechanisms because they can clear the amyloid really well. But even if they clear the amyloid really well, the effect in the cognition is not very big. And this is probably because they are applying these resistance mechanisms in this phase when they already have cognitive decline. And we know that the amyloid and tau starts to accumulate 20 years before this cognitive decline. So this accumulation has led to a cascade of biological processes that have been activated that we are not stopping with these resistance mechanisms. So at this stage, probably we need to combine resistance mechanisms with resilience. So that's why also it's important to try to understand these resilience mechanisms. So to study resilience, I started a collaboration with Teresa Gomez-Isla in Massachusetts General Hospital. She has been working on the neuropathology of resilience brains for a long time. And what we want to do it was to combine my expertise on omics and data analysis with the neuropathology to try to understand better the molecular mechanisms. So the first thing that we needed to do is to, define our population and how we are going to define resilience. No? So to define resilience, we have two axes, the one of the cognition and the one of the pathology. So we have two very clear groups that it will be the controls. They don't have pathology and they have high cognition. And we have also the demented ID that is in the other side, that they have high levels of pathology and low cognition. So the resilience will be individuals that despite having high pathology, they have high cognition. Usually here, how you define pathology can be different. Most of the resilience studies are done with individuals that are at BRCA stage 0 to 3 or 1 to 3, that it will be that you have tau more in memory regions, medial temporal regions, and more initial regions of the disease. While we have been focusing more on BRCA stages 5 and 6, that it will mean that you have tau all over the brain. So these are really rare cases. We found that only 1% of the population will meet this criteria of resilience. And for example, from the 1,500 brains available in ROSMAP, only 25 were meeting this criteria. So we used those 25 and we searched for other 25 controls and demented, matching demographics and comorbidities. And we tried to understand a little bit better that group. So the first thing that we try to do is like, let's start with no a priori hypothesis and let's take the transcriptomic. We take the postmortem tissue and we look on how the genes are expressing. So, and we compare how the genes are expressing in demented individuals and in resilient individuals. So we observed that there were many genes that they were downregulated in resilient individuals. And if we look to what were the enriched biological process of those downregulated genes, we were observing that all of these downregulated pathways were related to having lower inflammatory response in the resilience individuals. So despite having the same amount of amyloid and tau, the resilience individuals, they did not activate the immune response that usually we see in demented individuals. No? So we have many biological processes like apoptotic process, response to cytokine, inflammatory response, and so on. And what was interesting is if we look to the correlation of antemortem cognition with the global pathology, the level of amyloid and tau, the correlation was kind of modest. But if we looked the antemortem cognition correlation with the immune response, this correlation and effect was much higher. And if we look specifically to the complement, one of the biological process that happened during the inflammation, that is the signal of the microglia to start eating the neurons, the correlation was the highest one, meaning that the higher your microglia start to eat your neurons or synapses, the lower cognitions you have. We also look to the proteomics of these brains, and we observe that the resilience individuals, they have more synapses than demented individuals, probably because it's not engulfing this, the microglia is not engulfing the synapses. And this is a little bit what also Teresa has been showing in the different studies. No? So the individuals, usually the resilience that we see here and the demented, they have the same amount of pathology, the same amount of amyloid, and the same amount of tau tangles. But what is difference between them is the astroglia and microglia are activated, while in the resilience individual are not. So I mean, both the transcriptomics and the immunostaining here, they have their pros and cons. The bad thing about the immunostaining is that you need an hypothesis, because you need an antibody to test your hypothesis. The good thing about the transcriptomics is, even if it's more noisier and you see less things, is that you can look to the whole brain without any hypothesis, because you have the data of all the genes. And looking without any hypothesis, we are already seeing the same thing, that the inflammatory response is a big part in the resilience. But why don't resilient individuals activate the immune response present in people with dementia? No? Would they have the same amount of pathology, so why not? And for example, here, with Teresa, we have been doing expansion microscopy. Here you see the microglia really small, so you can expand the tissue. And inside of the tissue, you can see, for example, the microglia bigger, and also the amount of synapses inside of the microglia. And you will see that, obviously, demented individuals, they have higher number of synapses inside of microglia. But what it was interesting also is that those synapses are colocalized with tau oligomers. So for those of you who don't know the difference between tau tangles and oligomers, we have our neurons, and the neurons, they have microtubules that it will be like the skeleton of the neurons to have that shape. No? So the tau, it will be the glue of that skeleton. So during Alzheimer's disease, it will be hyperphosphorylation of tau, and the microtubule and tau detach from the microtubules that start to destabilize. So what is going to happen is that in your neurons, you will have monomers of tau. They will start to clamp together, that you will have oligomers. And then when they pack a lot together, you have the tangles. So resilience individuals, they have the same amount of tangles, but the levels of oligomers is lower. And this is a little bit the hypothesis of why maybe they have an activated immune system. These oligomers goes to the presynaptic zone, and in demented individuals, the microglia trying to eat these oligomers will also attack the synapses and kill the neurons. We also observe that we have less tau oligomers in the dendrites, not only in the synaptic zone. So trying to understand a little bit better why they have these lower levels of tau oligomers, we decide to look to the cellular and molecular mechanisms behind. And in this case, what we were using was single-cell transcriptomics. So previously, we were using transcriptomic of a bulk tissue. So you have the tissue and the gene expression. You don't know if it's on any particular cell or anything, because in that piece of tissue, you have a lot of different type of cells and so on. Now, for each individual, we have a lot of different cells, thousands of cells that we get, and for each cells, we have the transcriptomics. So we can use the gene expression to separate the cells, because certain cells that will activate, they do different functions, so we can see in the transcriptomics we can differentiate between cells. And we know that there are some specific genes that they only activate in some certain cells. So the first step that we do is take those cells, and based on their transcriptomics, separate in 7 different type of cells: the inhibitory neurons, excitatory neurons, microglia, astrocytes, and so on. And in this case, we are studying like the effect of resilience in all the different cells, but I will focus more in this talk in the microglia because we are seeing the effect of the immune system. So then what you can do is, for example, select the microglia, and again, based on the immune cells, because we also have cells from the periphery. And what you can do again is based on how those cells are expressing genes, you can subdivide in different states that represent different things that they are doing, these cells. So here, what you can do is, for example, take the anti-inflammatory cells and say, okay, do we have different in gene expression between demented and resilient? Or the other type of analysis that the people will do is a proportion analysis. Do resilient individuals have more of one state of the cells compared to another? So we looked to 440 brains, and we were looking if there was any kind of population that was promoting cognition despite of having high levels of amyloid and tau. And we found like there was this subpopulation that the people with better cognition will have more of this type of cells. We also validate in the 25 resilience and demented that demented almost did not have this type of cells, and resilience and controls have many of those. No? So we call this state a resilient promoting microglia. And we want to identify why in some individuals the microglia will become like more detrimental and will start eating the neurons, and in other cases will be more reparative and restorative for the neurons. So what we did, it was one analysis that is a trajectory analysis. So we know the microglia will start in a surveillance state, and based on the microenvironment, it can change what it's doing. So these different lines represent how the genetics of the microglia is changing to convert in different states. So what we want to do is, okay, the microglia will start in a surveillance state, and why sometimes it will convert to a resilient promoting microglia, and in other times it will take this other path to convert to a microglia that it will start eating the neurons. So here in red, we have the complement signal, that it will be the signal that it starts to eat the neuron. And we start to identify markers, and when you activate the complement will deactivate, for example, no? And if you don't activate those, the complement will go up. So we have a bunch of different genes that we have observed that are mostly expressing just in this resilient promoting microglia. And it was interesting that these genes, we look a little bit in the literature what they are doing, and they are different studies that we found, experimental studies with mouse models, that these different markers, if you can enhance them in mouse models, these mouse models will live longer with less cognitive decline and less pathology. So we know that these markers individually are able to protect against Alzheimer's disease. So imagine if you have more of one type of cells that can enhance all these different markers, that it should be able to protect a little bit more. So what we do is computationally try to estimate with all the data that we have different compounds or different changes that we can do to promote this type of microglia. So your brain will have more type of this microglia. And this is just one silly example, but for understanding how we work. So these are, for example, vaccines. And so we have databases with different vaccines, and you will have that vaccine, what changes in the expression of the cells will make. No? So, for example, we take our microglia, which genes are expressing, and we see, for example, that the influenza vaccine, it generates something that is very similar regarding expression of genes to having the influenza vaccine. So we observed that there were two vaccines specifically that promote this kind of expression in the brain, no? And it was the influenza, and it was also the tuberculosis. And also we found last year they have published that, for example, in a really big cohort during many years, like, for example, how flu vaccination and all these kind of things can reduce the risk of Alzheimer's disease, and potentially maybe because it's training in some way the immune system and to be more ready to enter in this kind of stage. No? So we are trying now, we have many different potential targets, and what we want to try is to to test them and see if we can generate these brains to have more of this type of microglia. No? So with the help of Rosario Sanchez Pernaute, that they are doing brain organoids on ALS and also in Parkinson's disease, we are starting to do brain organoids of Alzheimer's disease. So here the idea is you can take the skin biopsy of one patient of Alzheimer's disease with ApoE4 carrier, and we can isolate the fibroblast, and from then we can reprogram them to convert in induced pluripotent stem cells. And this we can use to generate brains. So if we have the skin biopsy of a patient with ApoE4 that develops Alzheimer's disease, this brain organoid is going to start accumulate amyloid and tau plaque, and is going to resemble the disease progression. We are planning also to do the gene editing, so we will have the same individual with the same genetic, except that we are going to change the ApoE4 for ApoE3, so we have also a control. Now, with computationally, we are estimating different potential drugs that can assert this effect that we want on the brain organoid, and we will be able to test in different phases of the disease, different targets, and so on, how the brain organoid behaves. The other thing that we are starting to do also is spatial transcriptomics. So until now, the transcriptomics that we were doing was more focused on the tissue, but we didn't have the spatial location. So now what we want to do is see how different cells around amyloid or tau behave, especially in resilient individuals. The problem that we have here, you see the microglia. So previous techniques for spatial transcriptomic, the resolution was not very big. So it was very difficult to get the transcriptomics of specific microglia cells surrounding amyloid and tau, because you will get the transcriptomics of all this region that you could have more than one cell. But now we have the technology to be able to have a resolution of 2 microns. So here you can see the size, for example, of the microglia, and you can see that we can even measure the transcriptomics of the microglia at different points of the microglia. So we are also starting to do a spatial transcriptomics. Okay, so I have spoken about a lot about the postmortem analysis, but what about in vivo? Can we characterize the resilience and predict if these individuals are going to be resilient even if they have amyloid and tau? So what we are, so for defining resilience, what we do first is to check if they are amyloid positive or not. We take the amyloid PET, and or it could be the blood biomarkers of amyloid. And for example, having this amount of amyloid is correlated to having this pattern of atrophy in the brain. But there are some individuals that despite having these levels of amyloid, they don't have this atrophy. So we quantify the disparity between the amyloid and the atrophy to quantify how resilient these individuals are. And with these measures, we can predict really well longitudinally. In this case, we have data for 5 years, that the more resilient individuals, despite having high levels of amyloid, they will stay cognitively intact for longer compared to the other individuals. Here you have a little bit of everything. You have, we have taken AthNA4, so you have amyloid positives, you have some Alzheimer's. Most of them are MCIs or controls. Yes, for the resilience score, yes. And here we are correlating that resilience score with the cognition, with the PAC. And we see that it correlates really well. So despite, I mean, it's a better prediction than that having, for example, correlate amyloid with cognition, because there are individuals with amyloid that they don't have cognitive decline. This way, if it's the disparity, the correlation is stronger. And also we can use the plasma or blood biomarkers. In this case, we also see that you can see the same pattern of inflammation in the periphery. So it could be also a good biomarker of having amyloid but not having an immune response and measures for resilience. And we also observed that later there is a second resilience mechanism. So what it happens is some individuals, they will get a lot of atrophy in these regions. And what it happens is that those individuals that activate, well, you start to have a lot of atrophy here. If you are able to activate this region in the dorsolateral prefrontal cortex, it will create an alternative connection with the dorsolateral uh, so, um, with the frontoparietal network, and usually this will delay the cognitive decline for 2 years. And usually the people that are able to activate more this, despite having atrophy here, are the people that have like higher education and so on, that are creating an alternative connections in the brain. But this kind of protection usually is just for a couple of years. It doesn't last longer, usually because you are a really late stage of the disease. Okay, so I have spoken a lot about resilience, but it is important also, as we were saying, the resistance mechanisms, no? Because it's not only, I mean, we still have a lot of pathology, so if we can quiet the inflammation, it's not like taking out the inflammation, because we still need like the good inflammation, but at least the bad one, to try to control it and to guide the inflammation to do more restorative work. But we also want maybe to remove the amyloid and tau, or be able to stop the progression. No? So that's why we need to understand also the resistance mechanisms. The problem is to study resistance is more complicated than studying resilience, because how do you know if an individual is resistant? Because by definition, it shouldn't have amyloid and tau, but it doesn't have amyloid and tau because it's a control, or because it's a resistant individual. So what we are trying to do is use genetics to identify which individuals have a genetic predisposition of having Alzheimer's disease, and see so if you have a predisposition and you don't have tau and amyloid, we will define you as resistance. So what we are using is, until now, they have been identifying 98 different genetic risk loci that increase the risk of having Alzheimer's disease. For example, here we see ApoE, so the size of the circle will mean how much it increased the risk of having Alzheimer's disease. So we see, for example, ApoE, 20% of the population have ApoE, and is one of the biggest risk factors for late onset Alzheimer's disease. But then we have, for example, similar size TREM2 mutation. The only thing is that it's really rare in the population. Just 1-2% of the population will have. And then we have many other genes that their effect size is not very big, but are frequent in the population. And the thing is, you are not going to have just one mutation. You are going to have a lot of these mutations. So the idea is this polygenic combination can predict the amount of amyloid and tau we have. So if we take, for example, ApoE for individuals, we see compared to non-ApoE carriers that have way more amyloid, way more tau, and a lot of more atrophy. But if we do a polygenic combination of the rest of the genes that are not ApoE4, we can also see that they have an important effect on amyloid accumulation and tau. Maybe not as big as an ApoE4, but it's still sufficient to develop a cognitive decline. No? And we can see this with age. No? So ApoE4 carriers will be the red ones. So from 55 already, they have way more amyloid and way more tau compared to the rest. And but we see also that, for example, having others like lower effect polygenic combinations also leads to having more amyloid and tau later than 70s, for example, or 65. And it happens the same with cognition. So so we are seeing that genetics can predict really well how much amyloid or tau you are going to have, and it can be a way to define which are the resistant individuals, to try to identify them and see what are the molecular mechanisms protecting them. So what we were doing, it was like, okay, we have the BRCA stages, so let's take the genetics of each individual, and we can define at which stage of the BRCA stage they are in the disease progression based on their age and genetics, and see if they have lower pathology than they should have because of genetics and age, and based on that, we will define that they are resistant individuals. What happened that when we were trying to do this, it was a total mess. We were finding like really strange things and so on. So we tried to do a model to estimate disease progression. And in this model, what we observe with tau progression is it doesn't follow that well the BRCA stages. 50% of the individuals, yes. But in other cases, we were seeing that they were like 4 subtypes of accumulation, that the tau could start in any of these points. I mean, this is the typical Alzheimer's disease, that it will start in medial temporal regions and go to the lateral regions and so on. But we also have different subtypes that they do not accumulate in medial temporal regions, that are these 3 ones. That is the other 50% of the sample, no? And we have some of them just in the occipital regions and so on. So when we did the disease progression, we observed that it can start in any of these, but then it will progress the same way to the rest of the brain. So we were able to computationally define the skeleton of tau progression. So, and the tau could start in any of these 4 networks, and it will use these pathways to propagate. And we observed that there were some regions of the brain that were particularly spreading more tau, or when they than others, no? That they were these ones that we identified here. So the thing is, okay, can the genetics also predict which subtype you will have, or how this tau is going to progress using this skeleton? Because we have a lot of variability in Alzheimer's disease, a lot of variability in cognition, in the age of onset, in where tau is going to start, and so on. So we were able to isolate the effect of each of the genetic variants increasing the risk of Alzheimer's disease. So for example, here we have the ones that the main effect is in the medial temporal regions. For example, we see ApoE that it will have an impact in the whole brain. But for example, if you have these mutations, you will have higher chance of having in the medial temporal region spreading through this pathway to the frontal regions. While having maybe other mutation like these ones, it will lead to having more tau accumulation in posterior regions and so on. So looking to the combination of all the genes, we can identify better how is to progress tau. And with this, what we are trying to do is make better models of personalized predictive modeling, where we can subdivide the individuals, the Alzheimer's disease, based on different subgroups, study their disease progression separately based on their genetics. And we are thinking that maybe different therapeutical targets will work better in different regions of the brain, because we know that there are some genes that they only express in certain parts of the brains and not in others. So this way we want to identify these mechanisms looking to resilience and resistance individuals, that they are in different substages. So how we do this? So this is a quote from Francisco Lopera that it's in Spanish, no? That it means that we already have the cure in the nature. We just need to look to the nature, no? There are a lot of individuals that they have the genetic predisposition of having Alzheimer's disease, but also they have genetic markers that they protect them. So the only thing is we have to search which are those genetic mutations leading to protection. So what we are doing is we have huge databases with cognition and genetics. So what we are doing is, okay, we now know that we can use polygenic combinations of different genes to predict the individuals how much amyloid or tau they will have. And we also have the cognition. So we can identify those that they are performing better despite having a genetic predisposition to have amyloid and tau. So this way we are identifying many genetic variants. The only problem of doing like this is then we don't know if these genetic variants is protecting you because you don't accumulate tau and amyloid when you should, or because you are still accumulating tau and amyloid, but you are a resilient individual, no? So then what we take is our databases that we have with PET and genetic data. And for example, here what we are doing is I'm showing you 2 different genetic protective variants that we found. Obviously, ApoE2, everyone knows about it. So here what we took was individuals at high genetic risk of having Alzheimer's disease with ApoE2 and not ApoE. And what we see is that those individuals with ApoE2, they have less amyloid in these regions compared to the individuals that they don't have with the same risk of having Alzheimer's disease. For example, with this other variant, we are doing the same, but they don't show any difference in the amount of amyloid and tau, meaning that both they are cognitively better having this protective variant. But one is because it's making your brain having less pathology, and the other one is because it's able to cope with the pathology. So we also validate that postmortem. We see the same effect with ApoE, lower pathology, in this case, no, and both they have better cognition. So for example, when we are looking to all the genetic variants that do not show any effect on the pathology, but just in the cognition, we see in all of them more or less the same effect, less immune reaction to the amyloid and tau. While we are now trying to see different resistance mechanisms that are a little bit different depending on the mutation. And we are starting to explore a little bit more, but we see things related to more metabolism, more mitochondrial, and so on. But we're still exploring a little bit the potential implication. And looking to the resilience, what we are finding is like we will have genetic variants that are protecting again, like all the brain, while there are other genetic variants that maybe only protects of having tau in the precuneus, and from the precuneus to spread to the rest of the brain. So, this also, identifying this is also interesting because, for example, stopping in the precuneus could maybe stop to the tau to go into frontal and posterior regions. We also found with the two cases of extreme resilience of the PCN1 mutation carrier or the Colombian cohort that they were not having tau accumulation in the precuneus and the entorhinal, despite having in the rest of the brain, and that was leading to a cognitive preservation and so on. So despite of some of them having a higher effect and others lower, it could be interesting also to explore those ones that they have lower. So what we are doing is we have a lot of data of postmortem data at different omics. We have also with PET imaging genetics and so on. So what we are trying to do is to integrate all this data to do predictive models computationally to define different targets that will promote resilience or resistance and try to test them with human organoids to be able to derive new therapeutical targets that hopefully will be more effective. So just as a summary and to finish, we have different protective mechanisms that could be effective at different stages of the disease and are important to understand the different mechanisms because maybe we will, depending on the stage of the patient, we will need to apply one or the other or a combination. The role of neuroinflammation in Alzheimer's disease, how the controlling the inflammation could stop a little bit or delay the cognitive decline. Also, how important is to take into account the variability that we see in the disease, and maybe genetics and things like that can help us define this variability. And then that multimodal integration of many different data modalities with omics, genetics, neuroimaging, and so on is able to provide us a lot of information of potential targets that we can use to mimic what resilience and resistant individuals are doing in the brain. So obviously I want to acknowledge like all the mentors that I have been having to put all this together, to Teresa Gomez Isla, Keith Johnson, and Jaquel, and Jorge in Massachusetts General Hospital. Now Jorge is in Yale, and also here to come back to Jesus Cortez in Biobizkaia, the computational neuroimage computational group. And thank you very much. And I am looking for collaborations and networking in Spain. So if anyone is interested in the work we are doing, here you have my contact and so on. And maybe we can do interesting things.
Thank you very much. Questions for Ibai? But, yeah, very short question. Of course, I mean, resilience, resistance is fundamental in when an individual is going to develop disease, when it's already present. But probably another force in the opposite direction is co-pathology. I don't know if you are considering co-pathology. For example, vascular pathology could be influencing inflammation and could be related also to the degree of how the disease develop once you have amyloid tau. Are you considering also co-pathology in your
And
models?
now in these moments, what we are trying to do is to at least the groups to be compensated for other pathologies. So we have the same amount of comorbidities in each group. But it's true that we want to consider them and we want to put them in the system. Also, it's true that, for example, we don't see with the neuropathology difference because by definition, we were trying to put the groups that they were having the same comorbidities, but when you were looking to gene expression, we are also looking to the vascular needs. I didn't present those results, but we also observed that the resilient individual, they have less vascular changes. Specifically, we are also studying how the microglia is interacting with the vascular cells, specifically pericytes and so on. And we are observing that obviously that communication is not the same and is not as impaired as in the case of demented individuals. But we have to start to incorporate it more, that kind of information.
I have two questions. The first one is only to test if I have correctly understood the first part of your presentation. The resilience depends mainly on the good inflammatory response and the ability to the neurofibrillary tangles and eliminate the oligomers of tau.
Yeah.
Correct?
Yeah.
The second question is about the different polygenic scores that are associated with different types or locations of the neurodegenerative, the tau deposits and so on. And have you ever thought that this difference may be due not only to the polygenic, to the genetic background, but to different etiologies of the disease?
Yeah, probably. Obviously, the genetic is not explaining everything. Also, we don't know the whole genetic background of Alzheimer's disease. These are just the ones that we have found until now. Maybe those subtypes that the people have not been focusing on looking to the genetic, they have also some genetic background that is specific to them. But obviously, there are many other things that are not genetic that could be influencing the having specific subtypes or not. But yeah, we have to incorporate all of those. We have to explore different contribution of epigenetics or other type of information that could lead to.
The data about the vaccine that you have presented may suggest some relation to infectious diseases.
So we did a study to see if tau was propagating more from the olfactory system as possible from an infection or more from the locus coeruleus to the medial temporal regions and olfactory regions. And we found that the spreading pattern seems to follow more the locus coeruleus toward, but it's true that the infections can lead to having more amyloid that can lead to having to the tau spreading more quickly. So we think that yes, that infections have an important role, and it will lead to having high levels of amyloid that will progress to tau. But we haven't focused on possible infections and so on, and we haven't studied that data. I mean, we don't have the data in if they have infections or not, so we were not able to control for those factors.
My last question is very short. Why did you place the reserve at the end of the neurodegenerative process or progression, taking into account that the main factors that generate reserve are
Probably at the beginning, yeah, that's true.
Very early.
But somehow it was because this activation of the potential network that you have for cognitive reserve was more active when you start to have atrophy in the precuneus and in the entorhinal. So we see that this region start to activate when you start to have, to compensate when you start to have atrophy in the hippocampus and the precuneus. That's why I put it at the end, because we were seeing maybe you create that circuit earlier, but with resting state, the functional activation of that region and hyperconnectivity we get once you have the atrophy in the hippocampus and precuneus.
I have a question related to what I was saying about the selectivity of the different composition of the genetic risk score at the beginning of the symptom, which I find really interesting. What is your hypothesis for that? Is kind of the gene expression is different, and this make a selective vulnerability of depending on where the genes are expressed, or it's a
Yeah, the thing is, like, also if you look to all these genes, the biological process that they alter is really different. Some of the genes will be implicated in more immune system, microglia function, astrocyte functions. Other ones are more related to metabolism. So you have many different biological processes that all of them having mutations in many of these different biological processes leads to Alzheimer's disease. So probably because Alzheimer's disease is a cascade of biological process, and maybe it doesn't mind where you start because it will continue to propagate. No? So, I mean, it should be interesting also to try to explore, depending on which biological processes are altered first, how this disease progress is going to progress and so on. But I mean, there are things that we want to explore, but we haven't explored that deeply yet.
okay. So you have an hypothesis of why they are helping you to differentiate the region.
No, not yet.
You don't know if they are expressed differently.
I mean, we have some transcriptomic data. Yeah, now we are what we are doing is we have transcriptomic of all the brain, so we are going to test how these different genes express in the regions of the brain, and maybe we will see, like, for example, ApoE is expressing more in the medial temporal regions, and other maybe more in the. I mean, for example, we look to the mutations leading to more tau accumulation in the occipital regions, and usually if you look to GWAS of having those genes, they are related to having visual problems. So obviously there are some links on how they express and so on.
Which genes?
The ones that leads to have more tau in the occipital regions. So we are starting to explore, like, okay, damage to these genes or changes in expression in these genes, what cognitive deficits you will have, and also how these genes are expressing in different regions of the brain. We are trying, at least.
Well, thank you. Thank you very much. I'm a bit, I have to understand the framework.
Yeah, maybe too many things.
Yeah, because we are very much involved in heterogeneity, in phenotypes, and I guess you are working with a model of heterogeneity that is multifactorial.
Yeah.
It is quite closer to the more classical models of vulnerability than to the more recent or previous models of prion-like.
Yeah.
Prion-like. So thinking on about prion diversity or strain diversity and so on. So
yeah, and then they follow that progression of.
You can understand.
The only thing is, like, the starting point is different, but then the progression, even if it starts in the occipital, it will propagate also to the rest of the regions. It's just the starting point. Yeah.
Different progression patterns can be understood in terms of different properties of transmission properties of strains or different vulnerability, genetic, for example, vulnerability of different cell populations to. I mean, I just trying to orient myself in this very, very complex framework. But I was trying to you were several points in your talk. The differentiation between late onset and early onset Alzheimer's disease is very radical. For me, it is very radical. And it is one of the reasons why it is so radical is because you find atypical cases mainly in early onset cases, not only, but how would you understand this?
In this case, we only have focus on late onset. We didn't take early onset information. So all of the data I'm showing at least is late onset.
because if you put this into the in play, I guess that you have to bear in mind also senescence factors. At least
One of the
Yeah.
biological processes that we got, I didn't show, but one of the biological process comparing the transcriptomics of the demented versus resilience was that the demented individuals, they will start to have more senescent cells compared to resilience, that it doesn't happen despite having amyloid and tau.
which are mainly related to immune and inflammation processes. So I was just trying to understand it, but thank you very much.
I think I put there more questions than answers, but right.
One technical question: when you define this this resilient resilient ratio, comparing the amount of pathology with the atrophy, you're using amyloid or tau?
So we are using more. I mean, you can do with both, and you can say
but
I
I
am
don't think it's
resilient
the
to
same.
amyloid. Yeah, so
don't
me
really-
neither. So we are
Yeah.
using mostly amyloid, also because we have more amyloid data. But
Yeah, I mean, we don't have
also tau is
but I
usually
think-
more related to having atrophy than
Yes.
amyloid. So
But the thing
probably
is,
with tau you will have
if
less.
I mean, amyloid, as you know,
Yeah.
doesn't give you an stage.
Yeah.
So it gives you,
Yeah,
you have it or not,
Yeah.
and so you don't know the stage. And
Yeah.
so it could be the possibility that we were discussing during lunch, that individuals with a lot of resilience, these professors of university that are starting with first they have a lot of atrophy, They have positive amyloid PET, but the degree of resilience is huge because probably they have been coping with the pathology for a long time, and then but they have a lot of atrophy, so it wouldn't be very resilient, and according to your
But
ratio, right?
depending where is the atrophy, because we have seen like some individuals, they have a lot of atrophy, but depending where, and if they were so depending, I mean, some of the things I cannot tell, but I will tell you later.
Atrophy. I mean, I see them.
But usually in the ones that we see a lot of atrophy and that they are resilient, they have a lot of hyperconnectivity of different cortical networks. So they are somehow compensating with for example, also if we look to CRISPR mutations, they have more gray matter. Even if they have the same amount of pathology, but they are doing functionally better, and they have higher hyperconnectivity, they have alternative connections in the cortex and so on. So, and I think like these high cases of Colombia, like Lydia and Relin-Colbos, they have a lot of atrophy. The brain was even smaller than maybe patients with Alzheimer, but the cognition was good. And there is a lot of hyperconnectivity of other networks there. So somehow, we don't know why, they have atrophy, but they still, like the neurons that they have, somehow they create alternative networks.
Yeah. Well, my point is related to the amyloid positivity, which doesn't give you a stage.
Yeah.
So probably it would be better to have some measurement of the pathology with more possibility to do staging, like tau PET, or probably some of the plasma biomarkers that maybe can help to make a.
The problem is in all the databases that we have, thousands of individuals with amyloid PET and genetics, tau is.
So what
Okay.
we can do.
Okay.
I have time for more questions. Okay, unfortunately. Thank you very much.
Thank you.